Now, let's continue to talk about the structures of the influenza virus. Influenza A virus is a segmented RNA virus containing 8 segments. The segmented gene can encode different proteins. Segment 1, 2, 3, and 5 can encode PP2, PP1, PA, and NP. M1 and M2 are encoded by segment 7, and segment 8 can encode non-structural proteins, NS. Segment 4 and 6 can encode hemagglutinin, HA, and neuraminidase, NA. These two are spikes on the envelope. There are 2 glycoproteins on the envelope. One is hemagglutinin, HA, and another one is neuraminidase, NA. Now, time to learn about spikes, HA and NA. HA is hemagglutinin. The HA forms a trimer and has several functions. Because HA can induce hemagglutination, we call this protein hemagglutinin. Hema means blood. Agglut means agglutination. To cause, to adhere. And in refers to protein. When we add influenza viruses into red blood cells of human, chicken, guinea pig, the viruses can bind and aggregate the red blood cells. The hemagglutination test could be used to detect the flu virus. Left shows the agglutinated mat, which is positive reaction. Right is the non-agglutinated pellet, negative. So when we detect the viruses using this plate, which reactions do you think refer to the positive? You are right, the right part of the plate. HA is the viral attachment protein, binding to salicylic acid containing receptors on host respiratory epithelial cells, and enables the virus to attach to host cells. Through HA, the virus can bind to the respiratory epithelium and then invade the cells. And HA has antigenicity. HA elicits the productions of protective neutralizing antibodies and binds to them. We can use hemagglutination inhibition test, HI, to detect the anti-HA antibodies. When we add serum into the virus and red blood cell, the antibodies in the serum could inhibit the agglutination. That is hemagglutination inhibition test. How do we use the test clinically? The test could be used to detect the specific antibodies. And when we have the specific antibody, the test can also be used to identify the subtype of the flu virus. Because of the point mutations of HA gene, flu virus undergoes minor antigenic changes, then can cause seasonal epidemics. This is antigenic drift. Changes in HA can undergo minor drift and major shift changes in receptor specificity and antigenicity. Shifts occur only with influenza A virus, and the different HAs are designated as H1, H2, to H18. So there are 18 HA subtypes of flu virus. Summarize the functions of HA. 1. Hemagglutination. 2. Binding to salicylic acid containing receptors. 3. Antigenicity. Neuraminidase, Na. The suffix ase tells us that it is an enzyme. Yes, the neuraminidase glycoprotein forms a tetramer and has enzyme activity. The Na is sialidase and cleavage the salicylic acid on glycoproteins. The Na enables new virions to be released from host cell. Look at this diagram. Cleavage of the salicylic acid on virion facilitates the release of virus from infected cells. Last section, we mentioned one group of anti-flu drugs, M2 ion channel blockers. Here is another group of anti-flu drugs, neuraminidase inhibitors, oseltamivir, Tamiflu, and zanamivir, Relenza. The neuraminidase inhibitors interfere with the release of progeny influenza virus from infected host cells. This process prevents infection of new host cells and thereby inhibit the spread of infection. The drugs should be used to treat patients with acute illness caused by influenza virus who have been symptomatic for no more than 2 days. Let's summarize here. Besides amantadine, the M2 ion channel blocker, the other antiviral drugs could be used for patients with flu, oseltamivir and zanamivir. They are neuraminidase inhibitors. Functions of Na. 1. Sialidase enables new virion to be released from host cell. 2. The enzyme catalyzes the cleavage of salicylic acid and an adjacent sugar residue in glycoproteins found in mucus. The flu virus have to go through our nose where there is a lot of mucus and pass the obstacle to go down. This action allows the virus to permeate mucin and escape from non-specific inhibitors. 3. Antigenicity. The Na of influenza A virus also undergoes antigenic shift, and the different NAs are designated N1, N2 to N11. Okay, as for the HA, there are H1 to 18, and NA, N1 to 11. Influenza A viruses are divided into subtypes based on HA and NA. For example, H1N1, H5N1, H7N9, etc. Can humans be infected by all these subtypes? Let's have a break and then talk about the human influenza virus and avian flu virus.